We've already talked a lot about B cells and T cells and their structures and the types of antigen receptors they make. But we haven't really talked about how to make them at all. And that's actually a really complex process. And so we're going to begin to discuss that here. This video is going to focus on B cell development with just kind of an overview of development in general. And then I'll make a separate video for T cell development. Um, on my YouTube page, there is already a video that basically is like overview of B and T cell development that kind of goes through both processes right next to each other. So you're going to have a couple of opportunities to see this material to hopefully understand the broader scheme of how this all happens. All right, so before we get too into the weeds with all of this, I just want us to remember the overall structure of B cell receptors and T cell receptors, because the whole point of development for these cells really is to create these structures. So in a B cell, remember the B cell receptor is essentially an antibody that is affixed to the B cell's surface. So that's the B cell receptor, okay? The T cell receptor, on the other hand, is basically like if I took one of these FAB units and stuck it on the surface of the T cell. So this would be the T cell receptor, okay? The B cell receptor has a heavy chain and a light chain, right? So I should have done this different, but that's the heavy chain. And that's the light chain. So it actually has two heavy chains and two light chains, right? Because here's 1, 2, and here's 1, 2, okay? The T cell receptor has an alpha chain and a beta chain, okay? It really doesn't matter. We can say this one is beta and this one is alpha, just the way they're drawn, okay? The beta chain is akin to the B cell receptor's heavy chain, and the light chain is akin to the B cell receptor's alpha chain, okay? And I tell you this because as we go through and make these chains in this video and in the next video, it'll be helpful to remember that this exact process is going to happen again and again and again, okay? All right, so let's get into actually making a B cell receptor. So the first thing we want to do is think about what we're trying to do when we're developing a B cell. And as I've already kind of alluded to, the most important thing about developing a B cell is creating the antigen receptor. And this can't just be any antigen receptor. It needs to be diverse. You don't want too many of the same running around, right? Because we need to be able to recognize a wide variety of antigen receptors. We also need to be able to get rid of any antigen receptors we make that are potentially self-reactive, because once they are released from the bone marrow into the tissue, we don't want them to hurt us, right? And then we need to be able to mature and basically promote those B cells that perform really well for us. So let's start by just thinking about how we're going to start with number 1, which is generating a diverse antigen receptor. So if we think about what a B cell receptor looks like, we have our heavy chain here, right, in blue. And then we have our light chain here in green, okay? The heavy chain is going to be made up of 3 gene segments: variable, diversity, and joining. The light chain is going to be made of only 2 gene segments: variable and joining. And they're going to come together to form this unique antigen receptor, okay? Now, we inherit multiple copies of both heavy and light chain variable, diversity, and joining regions from our parents. So one B cell receptor might have a heavy chain that's, say, variable 1, diversity 2, joining 6. and a light chain that is variable 3, diversity 2, joining 1. Whereas another B cell might have a heavy chain with this signature. And you can see that because we're using different variable regions, sorry, up here I made a mistake, there shouldn't be a D there. Because we're using different variable and joining regions in these two things, we have completely different antigen recognition sites, and that's going to create for a more diverse antigen receptor. So we need to learn how to make these recombinations so that we can make a functional antigen receptor, okay? So that's goal 1, and it's going to go through these various steps. And for this stem cell, we're actually talking about a very specific cell, which is known as the common lymphoid progenitor. The common lymphoid progenitor is basically the common progenitor for all lymphocytes. So that's your T cells, certainly, and your B cells, which is why we're talking about them here. But also don't forget, that's also your common progenitor then for NK cells, because they are lymphocytes, all right? So the common lymphoid progenitor kind of begins in the bone marrow, and we're going to go through some steps that basically tell it what to become into. But you can see that each of these steps, if you look here, has a different set of what is rearranging at which time to get us to a mature B cell. And this mature B cell is what's going to be able to leave the bone marrow and head out into the periphery, which is really the spleen and lymph nodes, where it will complete its differentiation and eventually become an antibody secreting plasma cell. All right, so the common lymphoid progenitor, basically it's in your bone marrow, and it actually interacts with the stromal cells within the bone marrow. These stromal cells are largely mesenchymal stem cells, and they're going to secrete growth factors like IL-7 and SCF, which is stem cell factor, and that's going to basically signal the CLP, or the common lymphoid progenitor, to stay in the bone marrow and to bind to certain adhesion factors like KIT, or ICAMs, and that's going to keep the association with the bone marrow very tight. And the growth factors, particularly IL-7, are really important for helping the cell maintain its progress through the bone marrow, because the cell is going to develop as it moves through the bone marrow until it's an immature B cell that can be released. Okay, recall on an earlier slide that I said that there were a lot of stages, right? So the first stage was the common lymphoid progenitor stage, and that's where it's moving through the bone marrow, right? The second stage is this stage, the early pro-B cell. During the early pro-B cell, we're going to have, I'm just going to create early pro-B. There we go. We're going to have random rearrangements that are going to occur spontaneously in the exon regions, okay? If these rearrangements, and this is only happening on the heavy chain is going to rearrange first. So what's going to happen is you're going to get random rearrangements that's trying to get one of the D exons to rearrange with one of the J exons, okay? So I want you to think of it like this. You've got all these V regions. They're not rearranging yet. You've got all these D regions. They're rearranging. And you've got all these J regions. They're going to rearrange, okay? So we're going to try to get this D and this J to come together so that they're rearranged near each other, okay? This is going to happen at both, on both chromosomes at this point. If this rearrangement is successful, the cell will move on to become what's known as a late pro-B cell. If it's not successful, if it can't make a D and a J region come together, then the cell will die. As the cell is transitioning from early pro-B to late pro-B, when one chromosome, because remember, this is all happening on both chromosomes, when one chromosome successfully does this, it basically shuts off rearrangements on the other chromosome. And that is known as allelic exclusion. It's essential to ensure that each B cell synthesizes only one heavy chain, because that's all we need for this process. We need one heavy chain variable domain and one light chain, making one antibody specificity. Except for some very rare mutations, all of the progeny of this B cell will have one antigen specificity, and that's because only one chromosome actually gets to complete this process, okay? All right, so we got D to join to J. Now it's a late pro B cell. So we're going to continue rearranging our heavy chain. So we have this DJ that we made in the previous stage. We're now going to try to get one of these V regions to come and join the D and J that we used before. If this is a productive rearrangement, i.e. it's able to successfully get like V1, D2, and J3 to come together on the first chromosome, it's going to move on to the next step. Let's say it can't do that. For whatever reason, it's not able to make the loop, make the cuts, whatever. It'll move on to the second chromosome, and the second chromosome will get a chance. If that doesn't work out, now it's out of genetic material, the cell will die. Approximately 50% of the cells in this pathway will die by apoptosis. This is kind of the default pathway in lymphocyte development. If it is able to do it on the second chromosome, it will move on to the next stage. All right, so what is the next stage? The next stage here is the large pre-B cell. This is actually the one stage in all of this that I kind of expect you to know by name. This is an important stage. So if it successfully rearranged the heavy chain into V, D, and J, okay, then it's going to pair it with something known as the surrogate light chain. The surrogate light chain is made of V pre-B, okay, and this is going to, and lambda 5. Okay, so it's V pre-B and lambda 5. So this light chain is going to try to partner with the heavy chain we made in these 3 steps. So in these early steps, we're making heavy chains. We're now going to partner with the surrogate light chain, and we're basically checking using this surrogate light chain to see if this heavy chain is capable of making a good pairing with the light chain. Can it pair with the light chain? If it can't pair with the light chain, then the cell might as well die, because the whole rest of this process is for making a light chain. And if we made a heavy chain that isn't capable of it, then there's no point in continuing, and the cell should die. Okay, now the surrogate light chain makes this a functional pre-B cell receptor, which is what's shown here. It allows for the B cell to determine if it made a truly functional heavy chain. It is expressed inside an endosome, inside the cell, okay? And that's important. It's within the cytoplasm of the cell. It also associates with 2 surrogate side chains, Ig alpha and Ig beta. These are actually going to stay with the B cell through its development, and they're kind of serve the same thing as the CD3 structure, the CD3 complex that I've talked about before, in that they're going to provide signaling to the B cell once the B cell is able to be signaled, okay? Now, when this pre-B cell receptor gets signaled, because like I said, it's functional, this large pre-B cell is going to proliferate massively. And this is going to significantly increase our diversity. And the reason is that we only made one chain. So now that one chain is going to be able, this one heavy chain that we made in the previous steps, is going to be able to be paired with lots of different light chains that will be made in the previous steps. So waste not, want not, right? We made one heavy chain, and now we're going to use it with lots of different light chains to create more diversity, all right? Okay, so let's say we made this heavy chain. It can pair with the surrogate light chain. The cell proliferated. Now we're moving into the small pre-B cell step. So here's what I was showing you earlier in the large pre-B cell step. We have our heavy chain that we rearranged. It paired with a surrogate light chain, and it got a good signal. So therefore it proliferated wildly. Now let's say we're focusing on this daughter cell, okay? This daughter cell is now going to become a small pre-B cell, and it's going to begin the process of light chain rearrangement. And I mentioned in a previous self-study that there are two types of light chains, okay? There's kappa and there's lambda. They're both functional, they're both capable. Kappa rearranges first, and there's kappa 1 and kappa 2, so 2 chromosomes. If at any point we are unable to make kappa bind with the heavy chain, then lambda gets a turn, and it'll try to rearrange the lambda on the first chromosome and then the lambda on the second chromosome, and try to make something that can bind with the heavy chain. If it can't, then the cell will die. If it can, then it will just make a lambda light chain antibody, okay? If it was able to make a kappa, then lambda never rearranges. That's kind of the long and short of it, okay? Now, this is what gets kind of tricky. If we look at a kappa light chain, okay? A kappa light chain has about 5 J regions, and it has several V regions, okay? I'm just going to draw some in. It will keep rearranging. So let's say we had V1 bound to J1, and that was not functional. It'll go back to that same first chromosome and try to make another. So now we're at V3, J2 and V4, J4, and it'll just keep going back and back and back until it has run out of genetic material, until it's tried all of the combinations and it doesn't work anymore. Because every time it makes a rearrangement, it makes a cut, and then that genetic material is lost. So when it runs out of genetic material, that's when it moves on to the second chromosome. And then this chromosome will try. And when it runs out of genetic material, it'll move on to the lambda first chromosome. If that runs out of material, it'll move on to the next. So that's what I mean by it'll keep trying. The kappa light chain will just keep trying to make rearrangements until it does it successfully or runs out of material to make a successful antigen receptor. So this is what I was trying to illustrate on the previous one. So you can see you've got all these V kappas, right, on your first chromosome, and you've got 5 J regions. So it tried to do this, and it wasn't a good join. Let's say it couldn't bind to the heavy chain. So then it tried this VK2 and a different JK, and it still wasn't good. So this is all happening on the first chromosome. And if this was non-productive, it would then move on to kappa 2, and if it went through all of that and was non-productive, lambda 1, lambda 2, you get the picture. Now, if it makes it from the large pre-B cell all the way through small pre-B cell, and it happens to make a successful light chain that is able to bind to the heavy chain, then it's an immature B cell. Now it's basically expressing IgM, because remember the heavy chain determines what type of class it is, and it's always going to make mu first. It's going to have the mu chain, and then it's also going to have a kappa chain, unless it had to go down to lambda, in which case it's going to have a lambda chain. This is now a fully functional antigen receptor. It is rearranged and recombined its VD and J on its heavy chain and its V and J on its light chain to make an antigen receptor that can be on the surface, which is a functional IgM molecule. So I mentioned that during this whole process that there are lots of different cuts, and that's kind of what's happening over here, and I don't talk too much about it, but basically you have these proteins or enzymes that make specific cuts throughout the process. And these are important because they actually direct what happens at each stage. And you can see when they show up and when they don't. So for example, your V pre-B and your lambda 5, they start showing up at about early pro-B. They're heaviest at large pre-B when we would expect to see them, and then they die off. They're not being expressed anymore. A really important one to know is BTK, or Bruton's tyrosine kinase. Bruton's tyrosine kinase is important for actually making the jump, basically, to creating a functional B cell receptor. So you can see it's over here where it's darkest, right? And patients who lack Bruton's tyrosine kinase are known to have a BTK deficiency, and this is also known as XLA, or X-linked A-gamma globulinemia. These patients are unable to produce antibody, and therefore they wind up with these recurrent infections, and I've listed some of them here. All right, there's one more thing that our B cell needs to do before it can be released out of the bone marrow and into the periphery, and that's alternative splicing. We're going to talk about alternative splicing again later when we talk about B cell differentiation and antibody production, but alternative splicing is also how the body knows that a B cell has successfully completed antigen receptor creation, okay? Now, here's all of the VDJ rearrangements that we just talked about in the DNA, all right? And then they, get made into RNA and transcribed, and that's how, we have this antigen receptor site. We're done dealing with this part of the molecule. We're now going to focus down here, okay? So remember, the heavy chain is made as either using the mu gene segments or the delta gene segments or the gamma or the alpha, depending on if you're making IgG or IgA or IgM or IgD, whatever. Okay, now, when you're doing alternative splicing, you're making a change at the RNA level. And this means that you haven't made a full cut. Any cuts that are made at the DNA level, that's going to be a permanent cut. But an RNA level is kind of like a halfway cut, because now we might be able to go back and forth, okay? So alternative RNA splicing determines whether a B lymphocyte will express surface IgM or IgD or both. So an immature B cell that just finished B cell development expresses IgM. Once it's been tested and we know that it is not going to attack tissue outside the bone marrow, it becomes a more mature B cell. It's not differentiated, but it's grown up a little. It's seen a couple things, and it's ready to meet antigen on the outside of the bone marrow and eventually develop into a plasma cell. So alternative splicing occurs based on gene products that encode for transmembrane expression or secretion, which is when we'll talk about it later, or for the Ig mu or Ig delta process. So you can see here, since we're not making the cut at the DNA level, we're going to be able to have some of the antigen receptors that are going to express IgD and some will express IgM. So literally just if this was my IgM, and then this is my IgD. Now, the important thing is that because we're not making any changes up here, we're only making changes down here, the antigen specificity here is exactly the same, okay? The only thing that's different is kind of the plate it's being served on, all right? But it still binds the exact same antigen, and this VDJ region is completely unchanged.